Okay, so it's a great pleasure for me to introduce Professor Ian Dryden from University of Nottingham. Professor Dryden is a world-recognized expert in many fields of statistics, in particular statistical shape analysis, statistics of manifold, object data analysis, topic of today. Professor Dryden received the PhD from University of Nottingham, working with Kanti Mardian, with which he has many papers. He has received many honors, including being elected fellow of the IMS. He received also the Royal Society Wolfson Research Merit Award. He has been in the US many years. Now he is since about a decade or so back in the UK, and we are really looking forward to hear his thoughts about shape analysis on dynamic object data. So thank you so much for your kind availability. We're looking forward to your talk.
Thanks very much indeed. And thank you very much indeed for the invitation. It's a real pleasure to speak. So here's an outline of what I'm going to be speaking about. So a bit of an introduction at the beginning on statistical shape analysis and object data. And then I'll focus on three applications which are fairly short. One to do with placenta, one to do with clamp loaders. I'll explain what that means a bit later, and one to do with peptides. So I know it's a very broad audience, so I'm hoping that it's not overly technical at least, and I will try to just get the main ideas across. So if you've got any questions, please don't hesitate to ask, and I think just interrupt. I'm absolutely fine with that. Right, so have a brief look back at the analysis of object data. So I have a book on statistical shape analysis, and the first sentence is objects are everywhere, natural and man-made, and it's of interest for all of us to be able to analyze objects, so various sorts. And I know this group is particularly interested in biological objects and organisms and the shapes, and this is an area of real long-standing interest that people have been interested for centuries, in fact. So looking at early days, Galileo had, I don't know whether you can see my mouse here, hopefully, had the realization that larger animals are not just scaled up versions of smaller animals. Their bones for the larger animal here has to be proportionally thicker to carry the extra weight of the animal. So there's a shape change going from the smaller bone to the bigger bone. Darcy Thompson's famous grids, he had lots of pictures where he was mapping from one species to another. And here's just a couple of fish species. And this sort of geometrical way of transforming one object into another to explain the differences is a very powerful approach. So by shape, I mean all the geometrical information that remains when location, scale, and rotation effects are removed. So if I have my fish here, if I move it in the coordinate system, the shape's not changed. If I scale it, the shape isn't changed. And if I rotate it, the shape isn't changed. So that's what we mean by shape. Now, I have been working in shape analysis for probably too long, for a long time, since the 80s. And when I started, there was a couple of very important papers that came out. One from David Kendall, very mathematical paper on shape manifolds and Procrustean metrics, and one from Fred Bookstein on a much more sort of practical biological type approach on shape spaces. And the two had very, very important ideas and were very strongly related. And these two buildings that you can see here, the left-hand side is University of Nottingham, and the right-hand side, where I am now, and right-hand side is University of Leeds, where I spent some time a while ago. So the the area has really developed hugely, and there are many books on the topic. Very theoretical one on the end from a colleague, Wheeling Lay, and David Kendall himself. And then there's Bookstein, and there's a variety of books. And we're going through the years, and many people have written on this topic. One of the first applications I considered was looking at mouse vertebra shape, and this was on the front of the first edition of the book. This is a T2 mouse vertebra, and the experiment came from where mice were bred and selected according to their body weight. So one of the lines was they were selected for heavy body weight, so really big, heavy mice after 8 generations or so were in one group, the large group. Another group, the smallest, lightest mice, were bred with each other over a number of generations, so they were really small mice at the end of the day. And then there was a control group where they were just allowed, to mix as they wanted. And on the top middle here, we see the outlines of these bones colored by the different groups. Large are red, blue are small, and black are the controls. And obviously it looks like a mess, but we need to get rid of the rotation and the translation. And on the right hand side, we can see they're sort of matched up using Procrustes analysis. And then once you've got rid of these rotation, scale, and translation, then we can do statistical analysis, for example, principal components analysis. So here's some scores, and we can see the groups are quite different, a tiny bit of overlap, but largely very different. And then we can attempt to describe the variability with the sort of grids that Darcy Thompson had. Right, now that was shape, but object-oriented data analysis is where the same ideas are used in a whole host of different geometrical, usually, or object type data. So the first question one needs to ask is, what are the data objects? So the object is like the most fundamental unit of our object-oriented data analysis. So we might have data on a manifold like a sphere, we might have brains, we might have functions, we might have networks. Here, these are little covariance matrices from diffusion tensor imaging, and here are some shapes of DNA. And the same types of approaches can be used in all these different applications, and had a book fairly recently, with Steve Marin, who is the father of this area of object-oriented data analysis. So we want to know, after we've decided what the data objects are, which isn't always obvious, what's the object space? So where do they live mathematically? Often we want to take some features for carrying out practical analysis, like the scores, PC scores, or something similar. And then we want to use methods in order to answer the questions that we have available. So the sort of things we want to do with data objects, like shapes, but more generally, first thing is, can we say how far apart two objects are, like a distance? Can we define what a mean is, like the population mean, sample mean? Can we carry out statistical tests for saying whether these means are different or not? Look at the variability, so principal components analysis or other methods. Looking at regression, can we build a regression model where the data objects are either response or the covariate? And so when we have the invariances under rotations or whatever we have, like phase warping is another, which we'll see in a minute, the spaces we're in become nonlinear, and these were called quotient spaces because we're quotienting out things like rotation, scale, etc. So here's a quick example of DNA. We've got some 3D DNA molecules here, and we need to get rid of translation, rotation, and possibly scale. which we do for shape analysis. So on the right here, these ones are all registered using Procrustes analysis. So that registration is really important to get rid of the things we don't want. And functional data analysis, here some famous growth curve data where we're looking at the change in height of some children, starting at like when they're a baby, and the change is dramatic, and then they hit a sort of a growth spurt when they're early teenager. Obviously depends on the sex of the child and other things. People hit that earlier than others. So if you're going to analyze the data, you need to match the curves using some sort of time warping or phase invariance. So that's another type of object data. And as I've said, there's a non-linear aspect to this usually. So when you're getting rid of scale or rotation or phase, then data live in a non-linear manifold. So something like the following picture, which is like a smooth manifold. Locally, if you look really closely, it'll look like a Euclidean space, but there's some curvature often. And a Riemannian manifold is where you have tangent spaces like this one here, which varies smoothly across space. Right, so I'm going to, as I said, go through 3 applications. So here's a chance to stop. And are there any questions at this point?
Questions?
Because it's hard for me to know whether anyone's listening. So it's always good if there's a question of some form. There doesn't have to be, but yeah.
Good. Everybody good? Yep. Yep. Good.
Okay. I'll continue.
Yes, please. Yes.
Yes. Sorry. Yeah. Okay. So the first application is some analysis of human placenta images. So the data actually, this is sort of an ongoing project, and really we haven't spent too long on it yet, but I thought it would be interesting to use it as an example. We've got the human placenta, we've got the maternal side over here, if you can see my arrow, and the fetal side over here, the umbilical cord. And so we've got images of the placenta over time, with where some mothers, expectant mothers, have they're put in the MR machine, and they have their images taken. Some of them many of them are healthy, and some are compromised pregnancies. So it's of interest to describe the differences that you see in the groups. So we actually get the data in a sort of rather it's simple form. It's just slices, binary slices, either in or out of the placenta. That's what we're given, and that comes from computer scientists who segment the images. And then it's of interest to compare the shape of the placenta over time. So this is a sort of dynamic shape. We want to see how the placenta changes. This is joint with Emma Zhan and Yushan Wu at University of South Carolina, and colleagues from Nottingham as well. So this is the actual data we have. So these are 2 views of the placenta, the sort of side view, which maybe is perhaps a bit more familiar, and also one from the top, essentially, in this view. So we've got 19 data points. The first 10 are 12 seconds apart, and then after that they're 120 seconds apart. So there's bigger gaps between those, and it's of interest to describe the change over time. I mean, they look pretty similar when you look at these, but there are some changes going on which we want to be able to describe. So this is a sort of OODA, object-oriented data analysis question. The data objects are, in our case, we've only got access to the segmented binary volume. So we've got these 19 images, which are binary, just slices. And we want to be able to consider the underlying object is is the placenta, the continuous path in time of the through the placenta volume space. And we're going to use this method that we call principal nested spheres, which has been around for sort of 15 years or so, which is a different sort of decomposition from principal components analysis. And I'm going to tell you a bit about that and then apply it to these data. So the methods we're going to use are fitting PNS and then looking at the PNS 1 and 2 scores to describe the main shape changes. So we have lots of choices really when we come to dimension reduction. We can decide whether to remove size or not. We can consider this PNS, this principal nested spheres, or maybe just linear methods like PCA. We can have different types of this PNS, which I'll tell you about in a minute, either great or small circles, or we could use something like a spline through space to describe what's going on. So our usual way of approaching a manifold dimension reduction is to look at PCA. So we would normally have some data here. There's just 3 dimensions, but often, and like you'd be in much higher dimensions for the placenta, huge, tens of thousands of points. And you will find the mean in the middle of the data, then look for the direction of greatest variability. That's the first principal component. And then the second one is the next biggest orthogonal to that. And then in this case, we just project down to 2 PCs to look at the scores after you've done that. So it's a sort of a forwards approach. Find the mean, then find the first PC, then the second one, which will give you a 2D space, the first and the second together, and then the third one that gives you a 3D space, and continue like that. So that's traditional method, been around for a long time, maybe nearly 100 years, and this is used very, very widely. So manifold dimension can be also done the other way, like a backwards approach. So if we had data on a high dimensional sphere, which is what this picture is here, and we want to reduce the dimension, one thing we could do is to fit a sphere which is 1 dimension less. So if you've got a sphere here in d+1 real dimensions on this side, and if you do a slice through it, then that gives you an actual sphere. Although it looks like a circle here, it's a sphere of 1 dimensional less. Okay, so that gives you a 1 dimensional less sphere, d-1 dimensional sphere, and then we can hop over to the next phase where we will then try and find another slice through the next dimension down to give a d-2 dimensional representation of the data. So we're trying to fit the data so that at each stage we only get rid of the smallest amount of noise possible. So we try and retain all the variability in these circles that you can see, the red circles here. And eventually you'll get down to a normal sphere that we're used to. So S2 in 3 dimensions. And then we'll fit a circle, like a great circle or a small circle. And then we'll finally fit a point. So that sort of transition through the dimensions backwards will give us, at the end of the day, a mean, principal nested spheres mean, and a first score around the circle, and then the next score will be the distance away from that circle. So this picture might help a bit more. Here we have some data which are down at the sort of penultimate stage on the usual sphere, and then we're trying to fit the best, in this case, great circle through the data. This is the green curve here, and then project on to there. Those are the first PNS scores. And if we do a small circle, then here this is the best fitting small circle, which may be overfitting a bit in this case, and the blue points are the PNS scores. So there's an alternative to PCA, which looks at, instead of linear dimensions, you're looking at circular ones. And when you have the great circle, they're not so different from PCA, but when you have the small circles, they can be quite different. And there are various methods for fitting this PNS. So in the shapes package, we've got PNS, and then another one when you have shapes available, and various backfitting procedures so that you can go from the score to an actual object and see what it looks like. So it's relatively easy, well, fairly easy to apply. Right, so let's go back to the placenta. So we've got 19 placenta objects with a large number of points on them, which is telling you about the surface and the volume inside as well. And the big problem in this case is that we haven't got a correspondence between, say, a point on here and a point on here. We need to estimate that correspondence. So it's not straightforward to do this, but then there are various methods to do it. We have used some procedures which involve kriging at some point. So we would predict from a set of points or a grid on one object, and then see where the points get sent on the second object. And this is part of our method for getting corresponding points on the placenta. So what we end up at the end of the day is, for each of the placenta, a grid. Basically, we have got a surface and a volume inside as points, and they all correspond. So at all 19 points, you've got a grid which has corresponding points. So meaningful correspondence across the whole of the placenta. And then once we've got that, we can do our shape analysis. So we've got thousands of points which correspond, and then we want to look at dimension reduction. So we're going to use the PNS approach. So we put in all these points, and then we will reduce the dimension to the first few scores. So one of the things that we do, because we've got rather big gaps between the last few points, we actually fill in with interpolation it's actually linear interpolation. We just fill in some extra points so that we've got a sequence of 80 in total. And now we can adjust that later, but that's what we've done for now. So here is the data. Here's the beginning of the placenta sequence. These are the first 10, right? And then the next actual data is here, the data points here, and then we sort of interpolate in between. And so the placental movement through the sequence looks like this. So it's not like just a straight line. There's some sort of change going on throughout the sequence. And on the left here, there's PCA. This is PCA of the surface points around the outside, and then the volume are all the points on the inside, which they look quite similar, the two plots here. And this is PNS here. So we've got the first PNS score going from actually it goes from this end. This is the beginning of the time, and then ending up here. And then the second one has a reasonable bit of variability where something's going up in the middle and then going up again. So this is our representation in the PNS space, and here is the PCA space. Right, so what I want to do is just to show a video. I don't know whether this will work or not. We'll see. Where we're going to, oh, right. I'm going to show you the first PNS score, and it's actually got some scale in as well. But let's have a look. Right. Okay. So these are 3 different views of the placenta. Okay. And then I'm just going to go through time through the PNS or scaled score, which you'll see in a minute. So the changes are not huge, but maybe if you look on this end, you can see some sort of change going on. So this is moving through the space of the PNS score, the first one. All right, and then we're going to do the second one. So that sort of was like what one movement, but essentially in this data, there's a contraction going on. So there's movement in the placenta over this time period of I think it's 16 minutes in this case, and that's the PNS1, and then the PNS2. Try to just get rid of that. Then sort of it's in a different sort of direction, so it's sort of I don't know, like gripping in almost. So I think let's do that again. So it's coming in and then going out, then in and out. So over this 16 seconds, then this is that contraction that you saw in the middle. So from the rather crude original data that we're given, we can have a smooth path through the PNS space. It's slightly different that we scaled it as well. So that's why it says PNC there, because we actually introduced the scaling as well. But it's a very, very similar idea. And then once we've described this change over time, like this contraction, then we can compare different individuals. But at the moment, this is just one that I'm showing there. Right. So, any questions?
Maybe a one.
Yes, good.
So I probably have two, but I'll ask one. So
Yeah, right.
in these two movies, so
Yes.
almost look like in a very rough understanding. The first component seemed like something like it would have been an exponential. The second component would have been something like sine or cosine.
Yes, 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 yes. I think so. If you look at this picture here, right?
Okay.
So yeah, what's happening with this first component is you're moving from, and how we've drawn it here, from the left-hand side to the right-hand side, and it's just coming down. And so what's happening is you're actually essentially moving around this circle. It's like a circular space. So you don't come all the way back actually. So it's like a part of a cycle, but not the full cycle. So and then the second one, you're exactly right that it's it is like a trigonometric function. It's like you're going at twice. The period is half the period of the first one essentially. Yeah. So I think that's a good point that you've made.
Got it. Got it.
Yeah. 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 So yeah, it's very good.
Thank you.
Okay. Any more or not?
Other questions?
So
Good.
So so I've had approximately half an hour, so there's about 15 minutes. They're fairly short, the other two, but I just wanted to sort of give you an idea of other types of applications. So this, and I actually might ask some questions as we go through, so just to increase the interaction. We've got some what are called clamp loaders, and these come from cryo-EM studies where we can actually essentially take some sort of image of an actual protein. This is a multi-protein complex, very big protein, and you can actually sort of construct it in its different, it moves around and you can see different positions of this protein. So we have 7 objects, which are the 7 states of this protein, and it's called a clamp loader, and it basically it helps to bind to when DNA is being repaired. This clamp loader helps in the process. So it switches between open and closed state, so it can bind and unbind from the DNA. So these 7, and these we've only got 7 of these. They're they're difficult to get. So we've got 7 states of a multi-protein complex, a big protein, and it has either open or closed states. And what we want to do is to sort of try and get a movie that we had for the placenta, but we want to stitch them together, but we don't know what order they should go in. Okay, so there's no time recorded here. There's just 7 states, and the protein should go from an open state to a closed state. So that's the task. So it's fairly similar in some ways to the placenta. What's much easier is that we know which points correspond, that we have the same atoms in each of these 7. So there's no need to match parts up or anything. That's very easy. But we have no idea about any time. So we know that there should be a sequence going from closed to open state, but we don't have any time. So we want to see if we can stitch these together. So this is an object-oriented data analysis problem. Again, 7 sets of atom coordinate data. There's around 3,000 atoms in 3D, and the correspondence is known. That, as I said, that's easier. And we want to consider a continuous path through the shape space. And we're going to use principal nested shape spaces, which is very similar to PNS, except we're taking account of the rotation of the molecule explicitly at each stage. We did also in the previous one at the beginning, we registered everything, but here we do a bit more registering at each stage. So we're going to fit this PNSS, and similar colleagues as what Charlie Lawton's the person who sort of motivated and gave us the data and told us about the problem, and colleagues from Florida International have also worked on aspects of this. So I'm showing the data again. Now this is where I'm going to ask you some questions. So these molecules are in the closed state or the open state, and we don't know which is which, but we can sort of see with our eye what we think. So do you think this is open or closed? I'm going to ask you a question, so I want an answer.
Oh, based on the fact that there is less space in between, I would have said close, but just
Yes,
randomly.
you're right. No, you're correct. Yeah. So, and the second one?
I think it's moving towards open, but it's not open yet.
No. So it's probably closed. Yeah.
Close.
And then third one?
But I would say open.
Yes, I think you're right. Fourth one?
Open again.
Yeah. Fifth one?
Close.
Yeah.
Close.
Oh, sixth one's not so clear, I think. Seventh one?
Open.
Yeah. That one you said closed, which is okay. Yeah, it may be. Yeah.
So these are stationary. These are stationary, right? So they are in a stable position, right?
That's right. We've only got them in these 7 positions, yeah. But we want to be able to construct a sequence that passes through near to them all, that makes sense temporally. That's the idea.
Good.
Yeah. So, I mean, one of the first things we can do is sort of cluster analysis and just look at the shape distance between them, see how far apart they are. So it really looks like 2, 1, and 5 are very different from 3, 4, 7, and 6. So 2, 1, and 5 have looked closed, and 3, 4, and 7 looked open, and then 6 sort of looks maybe a bit closer to the open ones, but it's less clear. 6, where that's the one which is more in between. When you do PCA or PNS, you get the following plot. So PCA, you've got 3 points miles away from the others, and quite a bit of variability in the second direction as well. And for PNS, we've got, they more or less lie along a straight line, but not quite. But the second one's a little bit out. But the 6th one here was the one that was a bit more doubtful, is sort of clearly sort of a bit more to the close than the open. And that's true also with the PCA, but the movement seems to be much more 2-dimensional here, whereas it's almost 1-dimensional here. So what we can do is to look at the PCA and the PNS in order to get a idea of going from starting on maybe well, it doesn't matter which end we start. We could start up at this end and then move along the horizontal axis here with just changing PNS1 only, and then plot the molecule at each point as we go along. Okay, that's the idea. We'll start up here and then plot the molecule as we go along, including in between where we haven't got any data, and just to see how the molecule moves. And then likewise with PCA, I can go along PC1 in a similar way, and then I'll go PC2 as well. So it's another movie. This one's
Can I ask you a question?
Yeah,
Another question here. So
yes,
my question related to these exactly these examples here. So when you do PNS,
yes,
so is it so it's just a surface, right? It's just a it's a circle. It's not the disk, right? It's a sphere. It's not the surface of the
yeah.
sphere, right?
So it is a very high dimensional sphere, yeah, and you gradually reduce dimension, and so the last stage here, which we're seeing, this actually PNS1 is a circle, even though I've drawn it out here. If you move along here, eventually you'll come back over here. Okay, so it's a circular variable here, and this direction actually isn't circular. So I think maybe when we see the movie, you'll see how it's circular actually.
Good.
Yeah, because what we're going to do, we're going to move from here across to here, and then we're going to go back again, and you'll see a sort of how and map it back to the original points, and the original points move in circles rather than straight lines, which you get with PCA.
Great.
So maybe that's probably the best thing to do. Hopefully I can find it. Right, let's get rid of this one. Right, so I'll get rid of this. There we go. Right, so this is sort of a bit slow, but you'll get the idea. So this is PNS, shape PNS, and this is the going through the from the positive to the negative PNS score, and it almost looks like it's dancing. Like the art, you can clearly see the arms sort of closing there and then opening. So open, sort of close in the middle, and then open again. So this is constructed from those 7 only, and just moving along in the horizontal direction. So I think you can hopefully see the sort of circular nature, the way these original points go around in circles, or sort of circular direction. And this is PCA, where things have to move along straight lines from the method, so it has to be linear. So that's the first PC, and it hasn't really, well, I suppose it's sort of almost closed up there, but it's got to move in straight lines. And then when we come to the second PC, we'll be able to see that that's you need the second one as well to see that it's really closed. So when it'll get there in a second. At yeah, 5 more seconds or so. PCA2. So that's not much movement, but that's really clearly closing. This clamp is closing. So this clamps around DNA, and again, they're moving in straight lines, these points, whereas they were moving in circles. And you only really need one PNS, whereas you need 2 PC scores. Yeah. So okay, so that's that approach. And I know that my time's nearly up, so I'm only very briefly going to spend like 2 minutes on the last one, just to sort of give you an idea of what happens when your data are very spread out. So right, so the final one is where we have shapes which can be quite spread out, and these it doesn't really matter too much about the application. We've got some peptide molecules, much smaller number of points here, but the key thing is we can on the shape manifold we can implement a spline. So we need to, if we've got data that's very spread out, we need to be able to map a tangent space across the manifold. So this is called parallel transport. So if you've got a tangent space at one point and you move along like geodesics, moving the tangent space along here and here and here, you'll end up with a tangent space down at the bottom. And the method of Riemannian splines, manifold splines, will what it does is it involves parallel transporting your tangent spaces, or a tangent space, towards a common tangent space. So if you imagine you've got some data really spread out on a manifold, and you map them all back down into a common tangent space here, so these geodesics become straight lines. And then when you've got the data in this space, you can fit a spline through it, so a curved spline, and then map it all back onto the manifold. So this was a very clever method of Jupp and Kent from 1987, and we've just adapted it a bit to the spheres here. But the difference here is that you don't just have to like have one path through the space. You can have a really quite dramatic changes in your objects, whatever you're studying, and then put them all in this tangent space, fit using the standard splines or whatever, regression models, whatever it is, and then map it back. So it's quite a broad and general approach. I mean, this particular application, we were just predicting in between spaces, and we could fitting a linear spline actually worked better than some of the other approaches. Right, so I've finished. Just some brief discussion. This PNS or PNSS is appropriate for circular or cyclic type variability, and I would argue in the placenta it really is cyclical. You've got these contractions going on. With the clamp loader, you've got again you've got this circular movement of the arms coming in. One can look at all sorts of other statistical techniques on these manifolds. The key is often to map it into some intermediate space which you can do statistics in, and then be able to map it back. So that the unrolling, unwrapping was one way of doing it, and using PNS scores is another. You work out your scores and then map them here and do your statistics in here. And then you'll have noticed they're quite high dimensional, these applications. So there are computational methods for doing what we've just shown, and many other manifolds you can do this. For example, compositional data on the simplex, you can map to the sphere and do it there. So I have finished. So here's some references, and thanks very much for listening. So any questions at all?
Yeah, let's thank Professor Dryden for a wonderful presentation. So are there questions?
Right.
I can start without, hopefully without monopolizing the Q&A time.
No, that's fine. Yeah.
So one of the things I want to ask you is, well, maybe the first thing is when you pass through the quotient space.
Yes.
Very early on, the quotient
Yeah.
space can be quite nasty. Is that correct?
Yes. Absolutely. Yes.
In
yes.
that case, then how do you do anything? Because when it's corners, right?
Yes.
So yeah, so more or less at the end, when we do all these analyses, we have some sort of smoothness assumption hidden
Yes.
here and
Yeah.
there.
Yeah.
But yeah.
Yeah. So, for example, with shape space, 3D shape space, the quotient space has got some very nasty points to it. There are singularities. So so the curvature will just blow up at certain points, and you have to assume that you're away from them. So, for example, if you've got a set of landmarks, they need to be lying in 2 dimensions less. So you need to have like collinearities. If you have your points lying in strange configurations, yes, you're going to have real trouble. So but you're away from them in all applications, apart from really constructed artificial examples. That's where the problems are. But you only have to move a little bit away, and it's all right. So yes, the curvature will change, but as long as you can have a tangent space and locally it's Euclidean, then you're all right. So I think some are worse than others, some of these quotient spaces, but the shape space is one that is known to be difficult, but only near singularities.
And
So you just stay away from them. Yeah.
And in that space, which is complicated, there might be some components in which maybe unbounded components, bounded components, different type of components. And so maybe at a certain point, you will need to have a combination of the methods we talked about. Because there can be some, the simplest case is some sort of a combination of, the principal components for unbounded components or most flat components.
Yeah.
While when there is curvature, you might need, some sort of extra nested sphere component and something like that.
Yes. Yeah.
Yes, and I'm wondering if something is going to go wrong at a certain point because.
Well, I think it may well do. Yes. So then it will depend on your application. So with the ones I've shown today, I don't think you're very near any big problems. They're not that different. You've got thousands of points, and they're all laid out around an object, and you're only in a very small part of shape space. Really, it's not that different from it's pretty well behaved. If you've got a smaller number of points and they're moving dramatically, like you've got a few points, like human movement type data, where you've got a few points on the arm and you're doing all sorts of movement, I think you would be in trouble with some of those. So then, you've got to think about probably a different coordinate system based on angles and in a way that is not going near these problematic points. So you probably need to do look at some different coordinates if you're getting into trouble with singularities and things.
Beautiful. So there's
Yeah.
questions from
Yeah.
the audience. So I will start with the first one. So the first one is, given the complex geometry of the objects, how do you first account for rigid body transformation and scale differences, namely rotation, translation, and scaling? Would this typically be addressed through an optimization framework that maximizes similarity between the registered model and the measured object? I will start with this one, but there is another one I'm following after. Since reconstructing 3D shape usually requires at least two views, could SSA contribute to directly reconstructing the underlying 3D shape rather than being limited to tracking the motion trajectories of corresponding points?
Yes. So, well, the first question then was about the registration. Yeah. So yes, like say with the placenta, you need to estimate a correspondence and then register. So using Procruste's least squares type method, and then you might need to re-estimate the correspondence again, and then hopefully, when you need to use some sort of optimization algorithm, hopefully you will eventually converge, but there's probably no guarantee that you would. Yeah, so that's what we do there. We get rid of the rotation and translation, and the translation and the scale are usually easy. It's the rotation that's more difficult. Yeah. So if you don't do the rotation, your first component will just be dominated by a rotation, and then what's interesting will be after that in the lower scores. So
the second one was on reconstructing. Since reconstructing 3D shape usually requires at least two views, could SSA contribute to directly reconstructing the underlying 3D shape rather than being limited to tracking the motion trajectories of corresponding points?
Well, yeah, so I suppose that's the sort of problem I'm not so familiar with, I think. So this is where you're tracking is it points on objects from different views, and then you're putting, because our 3D coordinates came from 3D images. We actually had the whole 3D volumes. So is this more to do with reconstruction from different views? I think,
Looks like that.
you can do, yeah. So using sort of projective geometry type analysis, yeah. So you can certainly adapt things, and you in that type of situation. But yeah, you get hidden points if you're not careful. So that sort of projective shape work could be needed there. I don't think I've answered the question. I wasn't quite sure what it was again. Was it?
So I would say
maybe I can see the question.
Yeah, it's on the side. It's in the chat.
Yeah, I can't see that.
You might have a button at the bottom of the screen with the blue. There is a blue bottom right.
Oh, yes, yes, I've got it.
This one.
Yeah, sorry, I'm not used to WebEx. Yeah. Okay. Thank you. Right. Oh, yeah, I think I did the first one. I think so, yes. I would say probably you need to use the this projective geometry in there, as not just shape analysis, but I think it can be helpful and used, and I know that people have done that, as definitely. So yes. we have limited number of points used by trying, but not the whole mesh. Oh yeah. Yes. So, well, we use this G mesh, which is really good for constructing the whole volume, actually. So that's the program we used for actually from the surfaces to the volumes. So it just does it for you, basically. So I think using, there's many people have worked on many related algorithms. So it's good to make use of what's out there. There are some nice tools. So it depends how limited the limited number of points is, I suppose. But yeah, it sounds an interesting problem. Definitely.
Good. Other questions? Maybe we have one time for one last question.
Thanks for the questions. Yeah, that's excellent.
Well, if not, let's thank Professor Dryden again for the wonderful presentation. Thank you so much for your time, for the interesting discussion. Thank you very much.
Thank you very much. Thanks very much for listening. Yeah, it's really good.
Thank you.
Good.